Welcome friends, welcome to Datta Biology classes. In the last episode we have almost completed the photosynthesis. There was a little bit late which we will discuss today, and that will be the end of photosynthesis. Now let us start with that. While we have discussed that RuBP, ribulose biphosphate, reacts with carbon dioxide to form a instable compound, 6 carbon instable compound known as diphosphorybutyl, and which immediately broken down into 12 pieces of phosphoglyceric acid, and thereafter 1,3-diphosphorybutyric acid, then PGI or phosphoglyceraldehyde was produced, and out of the 12 pieces, 10 pieces came back for the reproduction of RuBP, and 2 pieces joins to form glucose. Now here is the detail we will discuss today. Let us start with that one. So, we have produced out of 6 CO2 and 6 RuBP a unstable compound was formed known as diphosphorybutyl, which was 6 pieces, which I told earlier. These 6 pieces is broken down into 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 pieces of PGA having 3 carbon. And it has been seen that out of the 12 pieces, 6 carbon was having 14 C carbon. It was proved that carbon has been incorporated from gaseous form to solid form. So it makes no difference which carbon it is, but it has been incorporated. This has been proved. Now this PGA, phosphoglyceric acid, so it has got one phosphate in it. Again, by taking the energized compound, that is 12 ATP, 12 ADP. So taking this inorganic phosphate, it has become 12 pieces of 1,3-di-PGA, 1,3-di-PGA. That is 2 phosphate group is there by taking 1 phosphate. To make it activated, so this energy has been prepared. Now it is to be activated furthermore to produce phosphoglyceraldehyde from 12 NADPH to 12 pieces of NADP. Subsequently, the question may come where this phosphate was gone. So we will lose 1 phosphate here. Minus H3PO4. 1 phosphate has been removed. So it has become single phosphate. This is known as active acetaldehyde. That is 12 pieces of active acetaldehyde known as PGI or phosphoglyceraldehyde. Somebody says as G3P glyceraldehyde 3 phosphate, glyceraldehyde 3 phosphate, or phosphoglyceraldehyde, as you can say, as per your wish. Now, out of this, we have to continue the cycle because we have to continue photosynthesis. Because our main aim of photosynthesis is to produce carbohydrate, that is C6H12O6. Now, to continue the cycle, we have taken 10 pieces, that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 3s are 30, 30 carbon we have taken. Out of this, RuBP is 5 carbon compound, 1, 2, 3, 4, 5, 5 and 1 will come. So 5, 6, 30, that is 6 pieces. This 30 will come to, this 30 carbon will come here to produce 6 pieces of RuBP. But RuBP was not there because it had one phosphate group. So another phosphate has to be added. Prior to that, it produces ribulose monophosphate. Ribulose monophosphate, by taking phosphate from ATP, produces 6 RuBP. Ribulose bisphosphate or 1,5 bisphosphate. Previously, we used to call ribulose diphosphate. Whatever it is, maybe. Now, it can be written in 3 different ways on 3 stages. That is, this is known as, this portion is known as carbon fixation. Right, carbon fixation, like this. That in between one unstable compound was formed, and which ultimately can get converted to 12 pieces or 12 molecules of phosphoglycerolic acid. This phosphoglycerolic acid having one phosphate, by taking another phosphate from 12 ATP, it will produce 12 pieces or 12 molecules of 1,3-dipGA, 1 and 3. That phosphate has been added in 3 positions. Again, it has become activated, and now again, by reacting with the NADPH, it produces bigger or 12 molecules of phosphoglyceraldehyde, and in this reaction, phosphate group is removed. And now, after taking this 10 molecules of phosphoPGA, we will produce 6 RuMP, which ultimately produces RuDP. So this is known as carbon fixation. This is known as RuBP regeneration. And another one is left, that is known as glycolytic reversal. Everybody says glycolytic reversal. What is that? We will have to know it further, how it is. So we will start with this. I have completed the entire course of glycolysis in my respiratory year, but let us have a little reminder on that, so as to enable us to understand this glycolytic reversal. How it was there? It was glucose to program breakdown of glucose is known as glycolysis. So glucose, I told you that the formula is G square, F square, then 6 times P, P6. G square, F square, P6. Write in this way. Glucose, then another glucose, 6 phosphate, then fructose, 6 phosphate, then another fructose, 1, 6 bisphosphate. Then this is 1, 2, 3, 4, 5, 6 carbon, it splits to E phosphoglyceraldehyde and dihydroxy acetone phosphate. Then phosphoglyceraldehyde, it produces 1,3-dipGA. 1,3-dipGA. Then 1,3-dipGA. 1, 3. This was a reaction of glycolysis earlier. G square, H square, then 6 times P. 1, 2, 3, 4, 5, 6. Everything starts with P. Phosphoglyceraldehyde, 1,3-di-PGA, right? PGA 1,3-di, no problem. PGA, then 3-PGA, then 2-PGA, then phosphoryl pyruvate acid, then pyruvate acid. So this was the glycolysis part. That is how glucose is broken down. Here, we just reverse it. How? We have already completed, this is PGA, huh? This PGA, we have 3 PGA. We have completed. Then PGA to 1,3-di-PGA. 1,3-di-PGA, we have completed. Then 12 PGA, we have completed. Okay? Then we start. Now out of this rest 2 phosphoglyceraldehyde. This phosphoglyceraldehyde, here we have left over 2 phosphoglyceraldehyde. Clear? These 2 phosphoglyceraldehyde, this is a phosphoglyceraldehyde, but here is dihydroxyacetone phosphate, which can be reconverted to phosphoglyceraldehyde, P-gal, highlight P-gal, phosphoglyceraldehyde. This is a 3-carbon compound, this is a 3-carbon compound. Because dihydroxyacetone phosphate can be converted to phosphoglyceraldehyde by triose phosphate isomerase, we have written there. So these 2 P-gal, here 2 P-gal, these 2 P-gal, 1 P-gal and another P-gal, 2 P-gal produces fructose 1,6 bisphosphate. Here, this produces 6, 1, 2, 3, 4, 5, 6. 6, 1,6 bisphosphate. Because here was 1 phosphate and here in 3 position also, another phosphate was there. So they join, these 2 P-gal joins head by producing 1, 2, 3, 4, 5, 6. This P, let it be here. And another P, let it be here. So this is fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. So we have completed this one. Fructose 1,6 bisphosphate. Then fructose 1,6 bisphosphate will produce fructose 6 phosphate. Okay. So glycolytic reversal starts. Then fructose 6 phosphate. 6 phosphate, this will go. So 1, 2, 3, 4, 5, 6. Fructose 6 phosphate. So another phosphate will be removed. Minus PO4. Phosphate we have removed. Fructose 1,6 bisphosphate. Then fructose 1,6 bisphosphate to fructose 6 phosphate. We have produced fructose 6 phosphate. Then fructose will be with the help of isomerase enzyme, which I have told that with the help of isomerase 1, 2, 3, 4, 5, 6. Will be converted to glucose 6 phosphate. Again, they will use 1 phosphate to produce glucose minus PO4. Here we have added glucose to glucose 6 phosphate, we have added ATP. Again, this is because of isomerase. Again, 1 ATP was there in glycolysis. So we are using that. And this PGLD, 1,3 bisphosphate to this. Here we have not mentioned one thing, that here phosphoglyceraldehyde to 1,3-di-PGA, that is 2 position is there. Here 3 PGA is there. So here because of substrate phosphorylation, this S3PO4 was going up. So simply you write, if you know this one, you write in the reverse order, will come to 12 PGA. Just try it for 1 or 2 or 3 more times. So this is known as glycolytic reversal, the reversal. There ends the Kelvin cycle, that is carbon fixation, regeneration of RuBP, glycolytic reversal. This is the simplest thing which you practice it more and more. The more you practice, the more you will be able to keep it in mind. First, you have to keep these in your mind. G square, H square and P6. 6 times write P, and then try to write it one and again, so that you will be in the examination hall, you will be able to write. Thank you very much. This is the last part of the photosynthesis. In the next episode, we will come with a new chapter. Till then, thank you. Goodbye.